synapses. And the person who was studying synapses at that time was John Eccles, Sir John Eccles, who also won a Nobel Prize. When? 63, maybe?
Same price with Hodgkin Huxley.
I think. Oh, same prize with Hodgkin Huxley, 1963. So the conductance basis of the action potential, the conductance basis of the synapse. So Eccles was in New Zealand, okay, and Rahl told Cole, I want to work on synapses, and Cole said, you need to go to work with John Eccles. So okay. So they were doing synaptic integration in cat spinal cord motoneurons. Okay? These were difficult experiments. This is the kind of experiment where the dissection may take you half a day, 10 hours of dissection to finally get the system prepared in a way that you could record from motoneurons and do something on the leg of the cat. Anyway, he did his PhD research in the lab of John Eccles and Archie McIntyre. And I knew Casey Cole at the NIH, and he described to me a letter that he got back from Casey Cole after Rahl went to work with him. And Casey Cole says, Eccles says, this guy Rahl, he's really smart, but he's different than the rest of us. The rest of us will go off to lunch, and Rahl will stay at his desk working out integrals. Okay? He was doing mathematical physics of neurons. Eccles left for Australia, where he became a lab director, and Rahl finished in the lab of Archie McIntyre. So then after that, Rahl did a postdoc somewhere. He stayed as a lecturer at Otago in Dunedin, New Zealand until 1956, and then he came to the Naval Medical Center across the street from the NIH in that group, which had a very strong group of biophysicists. Casey Cole, Will Rahl, a number of other people who went on to become famous. This is a statement. Will Rahl pioneered biophysical modeling and computation for dendritic neurons with a focus on functional insights, testable predictions, and reinterpretation of experiments. With research collaborators, he demonstrated the functional importance of synapses on dendrites, predicted the discovery of dendrodendritic synapses, and identified the dendritic spine as a possible mechanism for plasticity. That means something to some of you. For others, I'm not going to be able to elaborate all of what that means. Okay? Now, where is my story? Okay, here's my story. Will was, I said, my mentor, and after my PhD in 1973, I came back to the NIH, and I took a position in the computer division. I stayed there for 2 years, and then moved over to the group where Rahl was. Okay, it was called the Mathematical Research Branch that was founded in 1957. It's now called the Laboratory of Biological Modeling. Well, this group, 1957, that means it's how old? 55 years old. It's probably the longest running group of doing mathematical biology or theoretical biology in existence. The head of the lab was John Herron. He was recruited from the Navy operation to head this laboratory. It was decided by some forward-thinking people. We're going to get a computer center at the NIH. We should have some mathematicians. Casey Cole was one of the people on the committee. And so they talked to this guy. He said, yes, I can do that, but I will not be in the computer center. I will not be a service operation for the biologists at NIH. We will develop theoretical biology as we see fit, which was a pretty bold statement to say. He was making his model after a group that was developed in Oak Ridge, Tennessee by Alston Householder, the very famous numerical linear algebra person. So anyway, that's what they did. They had some staff, so on. But Will always told me, why did most of these biophysicists fled from the Naval Medical Center because the organization changed. They got a new head. There's a very hierarchical arrangement, right, in the Navy. If the top guy decides we don't want biophysics anymore, people leave. All right, so Will said he had a talk with the new executive officer, and the talk was to try to tell him, okay, this is what I'm looking for. So he explained to Will, basic research is okay as long as it is not too basic. So that was a signal. It's time to go somewhere else. So fortunately, he came to the NIH. Did I want to say anything more? Maybe not. Okay. So Will came in, and he was beginning to develop his theory of dendrites, how dendrites worked. Before that, people didn't know what dendrites. We couldn't record electrically from dendrites. They were too small. We couldn't see them. Does it do with reaching out for food? We knew there were synapses out there, so there's some electrical thing, but what does it mean? So Rawls' paper in 1957 in Science was the first one to begin to draw attention, okay? This is a lesson. The lesson is, if you're recording, all the recordings people were made were from the cell body, not from the dendrites. So they needed to interpret these recordings from the cell body. You have to use the proper theory. The dendrites are cables. Eccles disregarded, he used a point neuron model to estimate the time constant of the potentials he was recording, the brief transient potentials. When you do that, you're ignoring the cables, and you get a smaller estimate for the resistance, or a bigger estimate for the conductance. That bigger estimate for the conductance means that the cable terminals are far away. So Eccles said, you can ignore the synapses out here. They don't contribute to the electrical life. Maybe for something else, but not for the electrical life. Well, Rawls came along and said, you got to use cable theory. Here's the equation you should use to estimate the time constant. And when he did it, it came out saying that those synapses are not so far away. Okay? So he was really upsetting the story of John Eccles, who was about to get the Nobel Prize, right? Eccles was a very dogmatic individual. Okay? He did not like this, Rawls upsetting his apple cart. Okay? So when Rawls was developing some of his later papers, or just after this, it was hard for Rawls to get his papers published. Okay? Probably because Eccles was a referee for some of these papers. Fortunately, Rawls was beginning to make good friends at the NIH, people that were on other or editorial boards of other journals, and some of his first theoretical papers after this were published, surprisingly, in the Journal of Experimental Neurology. That's a big shift in those days, early 60s, right? But that got him going. I'll just briefly tell you this. Am I going to tell you this? I'm going to tell you this. Okay? So the classical picture of neuron interaction is I've got these dendrites, nerve fibers from other neurons come, synapse on my dendrites, synapse on my dendrites. I integrate this activity. Here's my cell body. I send a spike down my axon. So the communication is axons synapsing onto dendrites, sending action potentials, impulses to other neurons along the axon. Okay? Rawl was involved around the mid-60s with Gordon Shepherd, a neurophysiologist, who had recordings from a part of the olfactory system of the rabbit. He had recorded the potentials outside of the neurons. These are small potentials, less than a millivolt. They're not like recording membrane potential, which is 100 millivolts. Okay? But that's the signals that Gordon Shepherd had. The time courses at different positions, here, 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 as a function of time, given that he stimulated a bunch of axons to send action potentials back up into the cell body, back up into the cell body, out into the dendrites. So they wanted to interpret this. So Rawl made his cable model of the neuronal dendrites and said, I'm going to compute the distribution of potential inside the cell, and I'm going to make sure they match, because I'm going to compute the potential outside the cell, given what's going on inside the cell, and I'm going to match these time courses. So they did pretty good for this time phase 1 and this time phase 2. They couldn't account for this time phase 3 by these back-propagating action potentials. So they scratched their heads. Apparently there's no axon connections between these cells and this other population of cells. These are excitatory, these are inhibitory. But the position of these cells, and knowing they knew that these cells got inhibition during this late period here, they made the hypothesis that the only way we can get these cells, the only way we can get these inhibitory cells talking to each other is if these dendrites sent a signal to these dendrites that turned their inhibition on, that came back. This was an extraordinary hypothesis to make, that there was synapses between dendrites. When they did that, they were able to match these recordings. At about the same time at NIH were anatomists that were looking very carefully at this region of the olfactory system. And these guys got together and said, oh my God, you have made a theory that accounts for some of the things we are seeing from anatomy. A synapse that goes excitatory in one direction and inhibitory in the other direction. Okay? So this is the first discovery of dendrodendritic synapses. They submitted this paper to Science, and the guy said, nah, we're not really interested in that.
So how they calculate the equation? Numerical solution? Or
Numerical solution.
And what about the numerical solution of 68 was analog computer or something?
No, this was digital computer, right? We had a card reader and a printer that was connected to the computer at the National Bureau of Standards, okay, as well as the NIH computer. Here is the notebook. Oh my God, this is my mother's birthday, August 24th, and my son's birthday. Okay? So here, this is thoughts about dendrodendritic synapses between granule cells and mitral cells. The late potential is a dipole, da-da-da-da-da-da-da-da. Okay, where does it say something about, it seems like here is a model whereby dendrodendritic contact between mitral and granule cells could account for these phenomena. It was their hypothesis. We're always keeping a notebook. We should all keep a notebook, okay? Keep a notebook. Mathematicians who get involved in doing simulations like this, they're not like physicists or experimentalists. They don't keep a notebook usually. They write down on little scraps of paper, oh, I use this parameter value, use this parameter value, and it gets lost in the drawer. How did I do that again? Right? You should keep a notebook. Okay. August 24th, 1964, March 15th, St. Patrick, no, that's not St. Patrick's Day. March 15th, 1965, we attended Tom Rees's 9 a.m. lecture on the histology of the olfactory bulb. The important point for me and Gordon is that he has now observed several cases that look like synapses from mitral secondary dendrites to neighboring structures, which could very likely be the spines of granule cell dendrites. They said, we got to get together, come on over to the office, we're going to show you what we got, so on and so forth. So they ended up publishing a paper together. But the paper about the modeling is 1968, the Journal of Neurophysiology, and I tell everybody in this field that's interested in cellular neuroscience, you've got to read this paper, okay? This is, for me, almost as powerful as Hodgkin and Huxley, because there's several biophysical steps to go through to understand this. You understand about computing extracellular fields, you understand a little bit about back-propagating action potentials, and putting, CNS, central nervous system, physiology together with the mathematical models. Okay, I'm going to stop pretty soon. So, let's see, this is where I came in a little bit, actually, okay? My story is here, okay? I started NYU, the Courant Institute, for a degree in applied math in 1967. Things were pretty hot at universities in America in the 60s, right? The Vietnam War was going on, the civil rights movement was still going, the sexual revolution was beginning, free love. It was an interesting time in America, okay? Well, I started in 1967 in September. In March of the next year, the guy who ran the draft to take us to the Army said, you guys studying for a PhD, you don't have a deferment anymore. Okay, so I thought I was going to get drafted into the Army. I got called to go take a physical in Brooklyn. I went, and we're filling out the paperwork, and the Army guy standing up, big Army guy standing up in front of us said, you know what? He said, if any of you guys think you're not going to pass this physical because you've got asthma, or you've got poor eyesight, or you've got flat feet, forget about it. You're going to pass, okay? So this got to be more serious. And so I said, geez, I don't think I want to go into the Army. So I started looking around, and I found out about the U.S. Public Health Service. It's a branch of the military, don't carry guns, they do research, they inspect ships on the coast to make sure we bring in no diseases, they treat veterans and so on. I got myself into the Public Health Service at the NIH. I knew absolutely nothing about biology, nothing. But I went to the computer center. He believed me, you need me. There was a lot of young people going in. It was a resource for them to be in the Public Health Service. So I came to the job, and the guy says, you're going to work in my group. He looked at my list of courses and stuff. I've been an engineer, undergraduate. Oh, you've taken some electrical circuit courses. You can work on the brain. Okay,
But he was right.
for the wrong reason. So the project was looking at recordings extracellularly from several neurons. Each neuron has a characteristic signature for its action potential recorded extracellularly. Remember, these are small signals. So you find some template for each one of these neurons. It might be maybe 5 different neurons you're recording from, and you plot the quantitative properties of that template in some space. So this neuron spikes occur in a cluster over here. This neuron spikes occur in a cluster over here. It's a problem of cluster analysis of data. I hated that problem, okay? After about 4 months, I said to the boss, I don't like doing this. This is not mathematical physics. Can't we find something else for me to do? He said, well, there is this guy in this other institute who does mathematical physics of dendrites, of neurons. Okay. So I went over there, and this guy was like amazing. He was doing this mathematical physics. He had this physical intuition. He didn't have a big group. He was a modest guy. He worked with like one person at a time. I had as much time as I wanted to spend with Will Rull. That's how I got into this. When I saw him solving problems using math physics, I said, this is what I like. He was making an impact on physiologists. He was telling them how dendrites worked, and they were listening. I remember going to a Gordon conference. Maybe that one I was telling you about. The next one. Will was there. We're going to breakfast in the morning. I'm walking with Will along, and somebody behind us says, that's Will Rull. I got this chill through my whole body. It was like, oh my God. It was a great experience. So my first presentation